Here we have part 4 of Special Senses, page 566 in the 9th edition. And we're taking a look now at more or less the overview of the structure. This can get very complicated, so stick with me here. Here's the hard palate. That's the basis of the nasal cavity. And then we have up here, we have the frontal bone, and we have the ethmoid bone actually piercing the frontal bone. Let's get a better focus on this. There we go. And so we have the crista galli coming up through. The crista galli is a crest of the ethmoid bone going up through the frontal. And then on either side, we have this flat structure here called the cribriform foramina. It's a cribriform plate, and it has lots of little openings in it for these bipolar cells. Here is cranial nerve number 1. It's this olfactory bulb resting there that's going to be doing the work of smell sensation. And never underestimate smell sensation, except the fact that our cognition, our thought processes eclipse so much of our sensory experience. We're very easily taken out of the present moment, but if we can quiet the mind and actually smell, listen, and see subtle things, that we can actually be very present for smell. We have about 10,000 different scents that we're capable of smelling, 400 genes capable of amplitudes of as little as 1 part per billion or 2 parts per billion, which is right up there with dogs, except dogs are not confused or, how shall I say, eclipsed by thought. And so they can use a much larger portion of their cortex, called the rhinencephalon, to actually consider what they're smelling. Okay, so that's our situation here. This is the superior concha, covered by skin, is now called a turbinate because turbinates cause the air incoming that we've aspirated to circulate and become humidified and warmed. And we're now considering odorants, or that is to say, the molecules, the aromatic molecules that are dissolved in the air. If you cook your food only in water, you miss many of these molecules, but if your food has any oily component, or shall we say the organic phase of the food, then we might actually get some richness to the aromatics in the situation. Here is a big close-up. So now we're actually looking at the very end of the olfactory bulb as it's resting. Looks like a big antenna, doesn't it? And it's not an antenna that makes us look like a Martian, because it doesn't stick out of our head. It stays inside, and it rests right here on the cribriform plate of the ethmoid bone on either side of the crista galli. And there's openings here, the cribriform foramina. Here are, look at these are the olfactory, it says here, sensory neurons. And they come up here and they connect with the tract up here. This is the olfactory tract, which sends the signals back to the brain. And notice that we're forming a little glomerulus right there. And there's lots of little astonishingly complex interactions going on there with individual notes of a chord of sense. Okay, so notice down here at the bottom we have a, shall we say, a slightly gelatinous area here because we have mucus and we have these very elongated stereocilia. Okay, these are the olfactory cilia. Here's the dendroid, it comes in. Here's the cell body. Here's the axon. It presses on. It connects up here. We synapse with the glomerulus. And there's very unique types of synapses that go on there. So we need to consider that as we come down here on the page. Humans can distinguish 10,000 or so odors. We have 400 different smell genes. Okay, so that's pretty significant right there. Here's our transduction diagram. Let's get that into focus. Okay, and so here we have the receptor for a particular molecule. Most of these are aromatic proteins, not all of them. There's quite a wide variety of molecules that become aromatic and dissolve in the air. Okay, and then we have this little sequence here of events that lead us to a second messenger molecule, CAMP. And in this case, we're actually going to phosphorylate a protein. Okay, so here we have little pumps that activate things. And we have a calcium, also second messenger process going on there. But we need to come over here now and take a look at this wondrous little pathway here, the olfactory pathway. They talk about the mitral cells. These are mitral cells up here in these big spaces. And take a look here at here's the glomerulus. That's the complex little nature of things right there. Here's the olfactory cell right here. Okay, let's see what they're trying to tell us here. They're trying to tell us that we have the mitral cells, which are second-order sensory neurons. They're going to synapse with the glomerulus. Okay, and they're going to be picking up the axons from these cells that are actually picking up the specific odorant. Okay, and so it says here that the same kind of receptor, let's take a look here, converge on a given type of glomerulus. Okay, so the glomeruli are specific. Each glomerulus represents a single aspect of an odor, like a note of a chord. Right, so you might hear people who are in the perfume industry talking about notes. Okay, different odors activate different subsets of glomeruli, making different chords. So that's how we get to the 10,000 different scents that are considered here, even though we have only maybe 400 notes or something like that make these 10,000 events go on. There's also these special amacrine granule cells, which are not pictured in that diagram that we were looking at. They release GABA. You might remember from last semester that GABA is an inhibitory molecule. It's actually used by opiates. It's the second neurotransmitter after the opiate neurotransmitter, and it causes inhibition. Right, so we can actually turn off mitral cells, which are the major pathway cells to the brain from the smell origins, so that only highly excitatory olfactory impulses are transmitted. So we have a very selective group. Also, I'm indicating to you that our brain is not, how shall I say, configured to interpret smell. So we have a very limited English language, and many other languages are somewhat limited, usually not as limited as English. English is really good for law, science, and thought processes, not so good for expressing sensation. So as we take a look here, the olfactory tracts are basically going to take the information from the mitral cell and go to the piriform lobe of the olfactory cortex. So that's pretty deep, actually. It's not all the way as deep as the taste sensation, but it's pretty deep there into the, how shall I say, temporal lobe as we're beginning to approach the insula. Okay, so it's sort of that space between insula and temporal lobe. We're getting all that information in there. That's for pure ID. Okay, and that's only one of the pathways. So that pathway goes directly, and also remember, this is the only sense that we don't entirely go to the thalamus. We learned last semester that all sensation is supposed to go through the thalamus before it gets to the cortex, and the exception to the rule is going to be the olfactory sensations. We also have a subcortical route here, which goes to the limbic system, right? And so here we have emotional richness. And so this is very deep stuff. Not only do we identify and figure out what's going on with that smell. Sometimes we get information about our safety from smell, but almost always there's an emotional component. We'll come right back to this.